Hello there, welcome to the World of Socinet, where your academic needs are met. Alpha Mo will try to put this book for you if at all. Today we'll be looking at a very, very interesting and sweet topic called control of gene expression in eukaryotes. First things first, we have to understand what gene expression is. Gene expression, from the name, gene expression, it means the process whereby genes in form of DNA are converted to their gene products, which are usually proteins. So they must be converted, genes or DNA can be converted to RNA and also finally to protein. This happens through transcription and translation. There's a conversion of DNA to RNA, specifically mRNA, is called transcription, while the conversion of mRNA to protein or polypeptide is called translation. What does all of this mean? It means that living organisms have a specific way of storing information, which is in the form of DNA or call it gene, and this information can be converted to what we see, which are the proteins or phenotype, what we see through this process of gene expression. So therefore, since this is a very important process, there has to be a way, or there must be control, because you don't expect information to be expressed or genes to be expressed when they are not supposed to be expressed, because it involves a lot of energy. And this is also not just important in eukaryotes, but also in prokaryotes. So therefore, this is necessary because it helps to avoid wastage of energy and also to avoid the wastage of proteins that are to be synthesized. And this can happen in different ways. It can happen during the stage of, and this control of gene expression can happen in different ways in eukaryotes. Examples include the use of transcription factors, as we'll see soon. It can be also happen in other ways too, like through chromatin remodeling, whereby the DNA is repackaged, and this repackaging prevents the expression of set genes or DNA. Other ways are also there, like post-translational modifications. After the proteins are produced, they are temporarily repackaged and they are less expressed or less effective until when they are required or needed. So now what we'll look at next is the concept of mitochondrial DNA. Mitochondrial DNA is quite important because the mitochondria is usually described as the powerhouse of the cell. Why? Because it has this important function of producing energy for the cell in eukaryotes. That's why this topic is on the control of gene expression in eukaryotes, because prokaryotes do not have organelles like mitochondria. Therefore, we have to talk about the eukaryotic cells before moving on to mitochondrial DNA. So mitochondrial DNA, from the mitochondrial DNA, is located in the mitochondrial matrix. The mitochondria has different parts. One of the crucial parts, which is the inside of the mitochondria, as illustrated, is the mitochondrial matrix, and it contains this mitochondrial DNA. There are a lot of features about this mitochondrial DNA. One is that it is double-stranded. It is circular as well. Circular like the genes found in, or chromosomes found in prokaryotic cells. And also there are other features of the mitochondrial DNA, which includes the fact that it contains about 37 genes. And these 37 genes have a unique role of producing 2 ribosomal RNA, 22 transfer RNA, and 13 proteins of the electron transport chain. And the size of the chromosome or of the DNA is about 16,569 nucleotide base pairs. So what have I been saying ever since? At the talk say the mitochondrial DNA is quite very important because it is found inside the mitochondria and helps in the exact function of the mitochondria, which is to produce energy. And one of the parts of the mitochondria that energy is produced is the electron transport chain. It produces about 13 proteins of the electron transport chain. So moving on. Mind you, not all the proteins found in the mitochondria are encoded by the mitochondrial DNA. Some others are encoded by the nuclear DNA. Please take note of this. And this is a clear distinction of the DNA of the nucleus and that of the mitochondria. The difference is that the nucleus has a linear DNA, while that of the mitochondria has a circular DNA. Please take note of this. And their location is quite different as well. One is located in the nucleus, while the other is located in the mitochondrial matrix. Please take note of this. And one unique feature, or one unique phenomenon about the mitochondria is its transcription and replication. Why is this important? It is important because it is not exactly the same as that of nuclear DNA, because there are different modules that actually explains how the mitochondrial DNA undergoes replication. One of the most important ones is called the D-loop module. D-loop or displacement loop. So the whole concept, the whole idea is that I told you that it's double-stranded. There are 2 strands. One of the strands is heavier than the other. The heavier because of the fact that the heavier one has more of purines like guanine, while the L strand is the other strand, which is lighter, or light strand, it is called so because of it is rich in pyrimidine, such as cytosine. Now, this model, this idea, or model now idea, because they get different ideas on how this process they happen. So one of the ideas now say the D loop model. Other ones are also there, like the Rittles. On the subsequent video, we'll look at that. But here now, we'll look at the D loop model. So the H strand starts replication, and the L strand then follows. So that's the concept about this D loop. And in this process, there's a D is some sort of a disc shape that will be seen when the process happens. That will be illustrated shortly. And this process now, there must be origin of replication. That is somehow similar to that of the nuclear DNA. There must be origin of replication. So it is different for that of the light strand and the heavy strand. The light strand has its own outside a region called the non-coding region or the control region. But for the heavy strand, its origin of replication is within the non-coding region, as we'll see shortly. So how does this thing work? First things first, we have to observe that initially, the origin of replication of the head strand is within the same point of the non-coding region or the control region. That is where it starts. When it starts, it will form a D structure, as I've seen here. A D structure. That D structure or D loop will elongate, will continue, and it gets to a point where the origin of replication is found for the light strand. Then it will form a kink where the RNA polymerase is displaced. And mind you, why is RNA polymerase there? From what we know of DNA replication now, we've seen in a different video that the DNA replication does not happen de novo. That means there must be something that it must start with. And the DNA polymerase that is responsible for DNA replication is a special kind called the DNA polymerase gamma. Take note. Then, and on that, two other proteins are important here, like the helicase. The helicase here for the mitochondrial DNA is called the twinkle. Why is it called twinkle? Because of the way it appears like stars. That's why it's called twinkle when you observe them. Then also there must be SSB protein to prevent the rejoining of the strands after being separated by the helicase. So that is that about those factors are required here. And if you look at it here, there must be a parent L strand and or parental S strand and parental H strand, and the ones that are formed are called the nascent L strand and the nascent H strand, and the RNA, short strand of RNA, is formed there, as seen here. So please take note. So the whole process of DNA replication cannot happen without there being a starting material, which is a short strand of DNA produced by the RNA polymerase of the mitochondria. Please take note. And that's the gist about the story. So they are formed, and after they are formed, they form their separate, and obviously the process of DNA replication is semi-conservative, meaning that the product will have one parental strand and the one nascent strand. So that's what is illustrated here. So this is the idea of the whole process. And mind you, the RNA polymerase cannot just bind. There are specific regions where they bind to. They include the promoter sites. You understand? They have promoter site of the heavy strand and promoter site of the light strand. And this are located in the non-coding region or the control region. Take note. And there are other factors that must be there for this whole process of transcription to happen. So this is aided by the transcription factors for the mitochondria, T-farm. So at this point, we would round off and we look at some questions. You can pause the video to look at the questions to keep yourself, bring yourself up to speed of what we have learned so far. So take note of all of the questions and try to answer them. If you have issues, go back, rewind the video, and you must have said the answers at some point. If you have more questions, you can reach us through our contact means. And thank you for listening. Thank you for watching. I remain Succinate. Don't forget to like, subscribe, comment. We're always here to provide you with good videos that will help you study. Thank you very much. God bless you.